Okay, well, thank you so much. It's such a pleasure and an honor to be here with all of you. So Paulinda, thank you for that kind introduction. As Paulinda mentioned, I'm an ovarian cancer surgeon, gynecologic cancer surgeon. But actually today I was writing about this for another event that Paulinda and I are going to tonight as to what inspired me to want to also become a scientist in addition to all this kind of clinical training that I did. And the answer was the patients who were impacted by ovarian cancer. I remember being a second-year resident at UCLA wards, and I would take care of patients who had battled ovarian cancer and had faced recurrence of disease over and over again. And it didn't matter what we did, how many surgeries we did, how much chemotherapy we gave patients. Sometimes it wasn't working. And that's really what inspired me to take an additional 6 years of training so that I could also train as a PhD scientist with the hope of really doing translational research aimed at helping lives of women impacted by ovarian cancer. And I'm very excited that we've made some very, very good progress. At the end of my talk, I'll share some of that progress with you. But given that this is ovarian cancer and gynecologic awareness month, I wanted to take this opportunity to just tell you a little bit about the different gynecologic cancers, how genetic predispositions can potentially increase our risks, and then how do we look at genes and impact of those in treatment of cancer. And at the end, I'll give you a little kind of description about the kind of work that we're doing in the laboratory to advance this, our sort of make progress in this area. About that. So we can go to the next slide. Okay, so here I just wanted to kind of show you a picture of, what do these cancers actually look like. So this disease that we call ovarian cancer, we've actually learned through much research that many of the tumors actually come from the fallopian tube, which is the organ next to the ovary. But you see here, this is a pathologic picture of a uterus and also ovarian tube that's been taken out. There is cancer, this is how it looks, and this is how it basically looks under the microscope. The cells just look a little bit more angry looking, the nuclei are a little bit bigger, and the pathologists make the diagnosis. But there are other cancers that women face as well, cervical cancer being one of them. Disease is found in the opening of the uterus, and that's really cervical cancer, and we'll talk about that a little bit. The human papillomavirus is the causative agent for cervical cancer, and we'll talk about how one can prevent that. And again, this is how it looks under the microscope. And then lastly, but not least, the most common gynecologic cancer in the US is actually uterine or endometrial cancer. And this is a cancer that arises from the cells that line inside of the uterine cavity. The incidence of this is steadily rising. And we think partially that's due to a rise in the rate of obesity, among other factors. And again, this is how the cells look under the microscope. But I think I was asked to talk about genes, so I wanted to really take one step back and talk about what are genes, basically. So the genetic code in all of our bodies is through DNA, and DNA is a double helix. It was a big discovery. And actually, a woman was very involved in this discovery, Rosalind Franklin, who was a scientist who worked with Watson and Crick. She did not get much credit for the discovery of DNA, but the images that she took were instrumental in figuring out that this is a double helix. Sadly, she passed away, likely from ovarian cancer, at a very young age as well, in her 30s. So she's a hero, and I have a picture of her in my office for all those reasons. But anyway, so DNA gets transcribed into a message, which is RNA, and then RNA gives rise to protein, which in essence are the building blocks of the cells in our body. And this is basically a cell. The cells then give rise to the tissue, and this is how genetic information is transferred. So how can the genes actually be changed, and what are these changes that could lead to cancer? Well, Paulinda talked about this a little bit. There could be hereditary factors. For example, we could carry a gene, or we could inherit a gene through our families. But there also could be things within our environment: radiation, chemicals, smoking, we know causes cancer. Viruses like the human papillomavirus can lead to development of cancer. And certainly just random genetic changes that sometimes happen and change the DNA code, and that could lead to cancer. And when that happens, changes happen sequentially. There's one change, and then it accumulates more genetic changes. There's a second change and a third change. Once the changes are sufficient that the body cannot combat these alterations, cancer can occur. So where in our bodies can these genes be altered? And I think this is really important to talk about. So again, there are germline changes, which mean that they're hereditary. They're basically changes that are in every cell of the body, and those can be transmitted through generations and can impact the entire kind of body. And the most common one that you've probably heard about is the BRCA gene that predisposes to ovarian tubal cancers, breast cancers, pancreatic cancers, in some cases melanomas. But also, random genetic changes can happen in our body. We call these somatic genetic mutations. These affect a certain area of the body, and certainly these genetic changes can also lead to development of cancer. So I wanted to take a moment to talk about who should get genetic testing. And this is very important because if we carry genes that predispose to cancer, that can increase our risk of developing those cancers. There are now preventive strategies and screening methods that can be utilized. So cancer in someone who's diagnosed at a young age, that's always a red flag saying that could there be a genetic mutation. The same person develops more than one cancer. For example, a patient has uterine cancer and then has thyroid cancer, for example. Cancers that impact both sides, for example, bilateral breast cancers, or having two or more immediate relatives with a cancer diagnosis. And in some populations, there is increased incidence of some of these genetic mutations, and we'll talk about that a little bit more. But how is this genetic testing done? I don't know if any of you in the audience have had genetic testing, but it's rather simple. You meet with a genetic counselor. They take a sample. It could be blood or it could be a buccal swab. The sample is submitted for sequencing. Again, in sequencing, what do they look for? They look for the genetic code through the DNA. And now we're really screening a large panel of genes. We've gone beyond just BRCA1 and BRCA2 as we understand more about genetic underpinning of cancers or cancer hereditary syndromes. And we at UCLA have pioneered this for years and years. We've been doing this, and we're at the forefront of it. So I wanted to now, and I'm happy to answer questions. I want this to be interactive, so please feel free to interrupt and ask questions. More than welcome to do so. But now that we've talked about the basics of genes and genetic predispositions, I wanted to get into genetic predispositions that can lead to ovarian or tubal cancers. Again, the most common one that we know about is BRCA1 and BRCA2. It's actually found in upwards of 15 to 20% of all patients who are impacted by ovarian cancer. And now it's standard of care. Any woman who's diagnosed with epithelial ovarian cancer should get genetic testing because the risk is high. And you see there the risk in BRCA1 mutation carriers, 44% increased risk of ovarian cancer in lifetime, BRCA2 about 20%. And again, what is the incidence of this in the general population? It's about 1 in 300 to 500, but it's as high as 1 in 40 in women who are of the Ashkenazi Jewish background. So important to know about that as well. And here are just some pictures to show you what is it that this BRCA gene does. And if you know what it does, it's basically involved in repairing this DNA damage. So it's not a surprise that if the BRCA gene is not working, genetic mutations can accumulate and you can increase the risk of cancer development. But there are many other genes that also predispose to ovarian cancer, some of which I've listed for you here, and we don't have time to go into details, but just so you're aware of them. So Palinda alluded to this earlier. The biggest challenge, one of the biggest challenges with ovarian cancer is that we do not have early detection tests. So though we do ultrasounds, we could do this blood test, CA125, neither of those tests actually detect this disease earlier. And this is a challenge partly because we just found out where the cancer actually arises from the fallopian tube. And again, I think there's more research that needs to be done in this area. This is one of the focus areas of our lab, which I won't talk about in detail, but this is a very important mission. But having said that, any woman who has signs and symptoms of abdominal bloating, discomfort, changes in bowel, bladder habit that are just abnormal persistently should see a gynecologist. They should do an ultrasound and should consider ordering this biomarker CA-125. But because we don't have these early detection tests in women who carry these genetic mutations, we recommend risk reduction surgery. And what does that risk reduction surgery entail? Well, it really involves removal of ovaries and tubes. And again, because the disease can arise from the tubes as well, we recommend removing both. More recently, we have started adopting a procedure called opportunistic self-injectomy. Having learned that many of these ovarian cancers actually can arise from the fallopian tube, so many times when women have surgery, for example, to have their uterus removed, we recommend that the tubes are removed as well as a preventive strategy. What about genetic changes that predispose to endometrial cancer? Well, there's a whole list of proteins that I've outlined for you that are involved in repair of DNA. Again, not a surprise. These are proteins that get recruited when there is DNA damage. They're called mismatch repair proteins and are involved in repairing the DNA, preventing accumulation of genetic changes. But in certain individuals, these genes can be mutated because of hereditary kind of genetic predispositions, or they actually can be altered within the tissue, for example, in the uterine canal, and that can lead to development of cancers, one of which is endometrial cancer. Individuals who carry mutations in these genes are about 1 in 300 people in the US, and it's called Lynch syndrome, basically, and important to know about because it can predispose to many cancers, including ovarian cancer, uterine cancer, most importantly, and some colorectal cancers. And so what do we do in those cases? I have many patients in my practice who come and see me. We screen them with biopsies of the uterine lining. I don't know if any of you have had a uterine biopsy or an endometrial biopsy, but basically it's an office procedure where we put a small side-lid inside of the uterus, we aspirate some of the cells, and then look under the microscope, make sure that the cells look normal, or see whether there's any evidence of cancer. And certainly, again, in those individuals, once they're done with childbearing, risk reduction surgery is recommended. I wanted to take a moment then to talk about HPV. I think this is really important because now we can prevent these kinds of cancers. So HPV is the human papillomavirus. It's a virus, and it's actually one of the most common sexually transmitted infections. It leads to many cancers, including cervical, vulvar, vaginal, oropharyngeal cancers, and also anal cancers. And there are subtypes of it, some of which are more dangerous, like HPV 16 and 18, and persistent HPV infection can lead to development of cancer. So how does HPV do that? Well, it's a virus, it's very smart. Here's looking at the cell layer. You can imagine that, remember we talked about the cells that are the building blocks. So imagine this is the cell layer of the cervix. The HPV virus gets integrated, it kind of hijacks the cellular machinery of the host and then can cause abnormal genetic changes. And it kind of gets rid of the normal regulatory pathways. Cells can accumulate and then cancer can develop. And actually now we can look at it under the microscope. Here's a cervical cancer sample. Those little dots that you're looking at is the HPV virus. But now we can prevent HPV, and we can do that through vaccination. And the vaccine basically covers a large subset of HPV viruses. These are the guidelines and recommendations. It was initially kind of offered through age 26, but this is actually expanded up to age 45 by the FDA. And it's a very simple series of vaccination. Depending on age, it could be 2 or 3 vaccines. And of course, we all know that by doing pap smears and HPV testing, we have significantly reduced the risk of cervical cancer. But it's important to know pap smear does not detect uterine cancer, and it does not detect ovarian cancer. So then the next part of the talk, I wanted to kind of touch base on, well, how do we use genetic information to guide treatment for patients? And our treatments have come quite far through genetic analysis of tumors. So there are three ways in which we can do this right now. One is the genetic testing of the patient to see if the patient carries a mutation that could make that patient more eligible for targeted treatments. We'll talk about that. We can also look at the tumor. We can do genetic testing of the tumor to see what kind of mutations are in the tumor. There are other assays like immunohistochemistry where we can just stain the tumor cells and look for presence or absence of certain kinds of proteins. So one example of this are a class of drugs called PARP inhibitors. You may have heard about this. This is now FDA approved in treatment of patients with ovarian and other cancers. But the important thing to know is that these PARP inhibitors specifically work in tumors that harbor mutations in the BRCA group. And how does this work? Well, cancer cells are constantly undergoing replication. The DNA has to repair. You give the PARP inhibitor, so it kind of blocks that process. If there is no mutation in BRCA, the BRCA proteins come in, they repair, and the cancer cell goes on to live. On the other hand, in tumor cells where there's a BRCA mutation, this repair does not happen, and then the genetic changes become catastrophic and ultimately leading to cell death, suggesting that really these kinds of drugs work in women who carry these kinds of mutations. And this is the current FDA approvals. It's for women who are diagnosed with ovarian cancer. They get surgery and chemotherapy, then they go on maintenance treatment. Again, those who carry BRCA mutations, either in their body, germline, or in the tumor, or patients who have experienced recurrence of cancer and similarly carry a BRCA mutation. There are other kinds of treatments that are coming to the front line, and one of them are a class of drugs called antibody-drug conjugates. You may have heard about this. I wanted to just give you a brief kind of update of how these work. Basically, the idea is that there is an antibody that to it has attached a chemotherapy. And this antibody can bind to a cell surface protein, and it can target the chemotherapy to the cell that expresses that kind of protein. So it's a more targeted delivery way of chemotherapy to the cancer cell. Tumors express this folate receptor alpha. They are, or the target for this antibody drug conjugate, they are eligible to receive this treatment. And it has been helpful in many patients. It certainly is not curative, but it provides another opportunity and another tool for us to help treat patients. Many of you may have heard about immune therapy, so I want to take a minute and talk about how is it that immune therapy works. Well, it works in certain categories of tumors, and if you recall, we talked about these proteins involved in repair of DNA called mismatch repair proteins, and when those kinds of defects exist, what happens, the cancer cell has multiple genetic mutations, but then develops what's called these neoantigens, and it expresses these neoantigens on the cell surface of the cell, and that allows it to interact with the immune cells in our body. But cancer cells are smart, and they utilize mechanisms like expression of this cell surface marker PD-1, which then binds, actually PD-L1, here's the tumor cell expresses PD-L1, that then binds to PD-1 on the T cell. And what it does, this is actually a normal mechanism in our body to prevent our own immune cells to attack us. But again, the cancer cells are smart and they hijack these normal mechanisms to kind of escape and evade the immune system. So when that interaction happens, you see the T cell, which is supposed to kill the cancer as our immune cell, is exhausted. But now there are medications, including antibodies, that can come in and intercept and prevent this interaction. And when we utilize those, the T cell is happy, it's activated. It can now attack and kill the cancer. So this kind of treatment does work in some settings, and it's been FDA approved, and I'm not going to, again, go into the details of these trials, because this is all very new data. It has been FDA approved for patients who have that specific genetic mutation in mismatch repair and have advanced endometrial cancer in combination with chemotherapy, and it does provide better outcomes. Also, it has been approved for women who experience recurrence of endometrial cancer and have that kind of mutation. So again, providing us with new, more targeted treatment approaches for treating patients. This marker that we were talking about, PD-L1, remember I mentioned that the tumor cells have it. Here it is. Tumor cells that have this brown staining, you can actually stain for it, and you can see it on these cells. This is a cervical cancer sample. And also there's new data to suggest that in patients with cervical cancer, when you add this treatment, if they have that expression, they can have better outcomes. And that's both in patients who present with advanced disease or in patients who experience recurrence of cervical cancer. So in a nutshell, that's kind of a brief overview. I know it was a tour de France. I hope what I said made sense, but this is kind of where we are. But I wanted to now tell you kind of how we're thinking about tackling these problems, and my focus area is ovarian cancer research. And these new treatments that I talked about, for example, this antibody drug conjugate, mirvetuximab, helps, but invariantly patients progress. So we need better treatments. We need treatments that will just attack the cancer from the beginning and get rid of that recurrence problem, which is really kind of my mission. So what are the challenges here? Well, here we're showing you kind of what the tumor looks like. And then what happens is the patient gets surgery, gets chemotherapy. Certain populations of tumor cells that have these capacity, again, remember these tumor cells are very smart and they kind of utilize mechanisms used by our normal cells, including normal stem cells of our body. We really think that there's a subset of tumor cells that have stem characteristics, and these expand with multiple cycles of treatment, and they can regenerate the cancer. Not only that, they also surround themselves with certain types of cells, including immune cells, that kind of support their growth, but also prevent the other active immune cells from attacking them. So our goal is really to sort of target the tumor at both ends. We want to target the aggressive tumor cells that seed the cancer. We also want to target the immune cells that support their growth and also prevent the other immune cells from attacking the cancer. So how do we go about doing this? So I'm talking about kind of a very interesting, exciting project. We've been working on this now probably for the past 3-4 years, but I haven't talked about it extensively. But this is a collaboration with immunology colleagues at UCLA. And I'm very lucky at UCLA that I not only have incredible clinical colleagues to work with, I also have basic science researchers who I can collaborate with and get them excited and interested in working on ovarian cancer. But in a nutshell, my colleagues Lily Yang and her lab have generated an immune cell called this sort of iNKT cells. She has basically developed a strategy where she can make large numbers of these iNKT cells. And on the surface of these T cells, she can also put a kind of what we call T cell receptor, or a chimeric antigen receptor that can target the tumor cells, basically. So we have been working with her team, and she initially was working in liquid tumors, but now she's more interested in working on ovarian cancer, together to kind of look at how to utilize these very powerful immune cells to target ovarian cancer. And here's actually a picture. You can see the tumor cell and the iNKT cell attached to it. And we have a lot of very interesting, cool data to suggest that this very powerful immune cell can target not only the most aggressive forms of ovarian cancer that are kind of enriched for these regenerative tumor cells, but they also can target the immune suppressive cells that surround them and can really work very effectively. We've done this through analysis, these studies through analysis of patient samples, and we utilize patient samples because ultimately we want to treat patients. So that's very important to show that we can target and attack these tumor cells in the laboratory. As I mentioned, sort of the next generation of these iNKT cells have a CAR, or chimeric antigen receptor, on top, and they can target specific proteins that are expressed on the tumor. Here we show you expression of mesothelin, which is something that can be expressed on the surface of the tumor cells. And again, we have very nice data to show that these very powerful effector immune cells can target specifically the ovarian tumors. And again, this is a collaboration between Lily's lab, and these are members of my lab who have been working on this project. So we're in the very early phases of kind of writing out a clinical trial because we would like to take this kind of treatment to the clinic and test whether it can help patients who are impacted by ovarian cancer. We hope to be able to test this first in women who are impacted with platinum-resistant ovarian cancers. We're thinking of a mode of delivery of these effective immune cells into the peritoneal cavity, and essentially this is a project that's ongoing. So we do many other different projects, but I don't have a lot of time to explain all of that to you today, but I wanted to talk about this part of the work. And with that, I'd like to thank you all for your attention. Thank you for being here. And of course, I want to thank Paulinda, and this is Paulinda's organization, who have supported us incredibly throughout the years. Thank you for doing that. To do research, you need research funding, and that's very important. Philanthropic support allows you to utilize the research in the most exciting and most promising areas without providing restrictions on utilization of those funds. We're very lucky to have been funded throughout the years, and these are other kind of funding sources, but I'm sure you know this, but I cannot overemphasize the importance of philanthropic funding, so thank you, Paulinda.